Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The late 19th century was a period of intense scientific ferment, a time when the very nature of disease was undergoing radical reevaluation. Dominating medical thought, the miasma theory posited that illnesses, such as malaria, arose from noxious vapors emanating from decaying organic matter, essentially bad air. This theory, deeply entrenched for centuries, shaped public health policies and influenced medical practice, directing attention towards sanitation and environmental control as primary preventative measures. Against this backdrop of established belief, the work of Alphonse Laveran emerged, challenging the very foundations of contemporary medical understanding. Laveran, a French military physician, embarked on a path of scientific inquiry that would ultimately reveal the true culprit behind malaria, a parasitic protozoan residing within the blood. His initial findings, however, met with considerable resistance, a testament to the power of established paradigms and the inherent difficulty in overturning long-held scientific convictions. Laveran's journey into the microscopic world of malaria began during his service in Algeria, a region plagued by the disease. Born in 1845, Laveran pursued a career in military medicine, a path that led him to the front lines of infectious disease research. Stationed at the military hospital in Constantine, Algeria, he encountered a relentless stream of malaria patients, their suffering a stark reminder of the disease's devastating impact. The unsanitary conditions prevalent in military hospitals and the surrounding environment only exacerbated the problem, creating a fertile ground for the spread of malaria and other infectious diseases. Driven by a desire to understand the underlying cause of this pervasive illness, Laveran dedicated himself to meticulous observation and experimentation. He had been profoundly influenced by Louis Pasteur's germ theory, a revolutionary concept that posited that infectious diseases were caused by microorganisms. Pasteur's work, which had already transformed fields such as food preservation and surgery, provided a conceptual framework for Laveran's investigation, suggesting that malaria, too, might be caused by a living organism. Equipped with a microscope, a relatively new but increasingly powerful tool for scientific investigation, Laveran began examining fresh blood samples from malaria patients. This emphasis on fresh samples proved crucial, as the delicate parasitic organisms were difficult to observe in older, degraded specimens. In the rudimentary laboratory of the Constantine Military Hospital, Laveran meticulously scrutinized countless blood smears, searching for any anomaly that might shed light on the mystery of malaria. His breakthrough came in 1880. Within the red blood cells of malaria patients, Laveran observed peculiar pigmented bodies, structures that he initially described with cautious objectivity. These bodies, he noted, were not present in the blood of healthy individuals, suggesting a direct link to the disease. Further observation revealed that these pigmented bodies were, in fact, stages of a parasitic organism undergoing development within the red blood cells. He witnessed the formation of flagellated bodies, motile structures that further indicated the presence of a living organism. Laveran's discovery was a radical departure from the prevailing miasma theory. He proposed that malaria was caused not by bad air, but by a microscopic parasite that invaded and multiplied within the human bloodstream. This hypothesis, while groundbreaking, flew in the face of established medical dogma and was initially met with widespread skepticism. The problem Laveran was trying to solve was the fundamental etiology of malaria itself. The accepted wisdom attributed the disease to environmental factors, specifically miasma. Laveran's solution was to identify a living organism as the causative agent, a paradigm shift that required challenging deeply entrenched beliefs. In his initial publication, Laveran detailed his observations with meticulous precision, providing detailed descriptions and illustrations of the parasitic organisms he had observed. He presented his findings at scientific meetings, seeking to convince his colleagues of the validity of his discovery. The response, however, was far from enthusiastic. Many prominent physicians and scientists dismissed Laveran's observations as mere artifacts, products of blood degradation, or irrelevant debris. The limitations of microscopy technology at the time contributed to the skepticism. The microscopes available in the late 19th century were often of variable quality, making it difficult for other researchers to replicate Laveran's findings consistently. Moreover, the preparation and staining of blood samples required considerable skill and experience, further complicating the process of verification. The miasma theory held considerable sway, and the idea that a microscopic parasite could cause such a debilitating disease seemed implausible to many. The scientific community was accustomed to identifying bacteria as the causative agents of infectious diseases, a consequence of the burgeoning field of bacteriology spearheaded by figures like Robert Koch. The concept of a protozoan parasite, a more complex and less understood type of microorganism, as the cause of malaria, was a difficult pill to swallow. Laveran's persistence, however, gradually began to erode the wall of skepticism. He continued to refine his methods, meticulously documenting his observations and presenting his findings with unwavering conviction. He understood that convincing the scientific community would require more than just his own testimony. It would require providing irrefutable evidence that others could independently verify. The increasing quality of available microscopes aided in this endeavor, allowing other researchers to examine blood samples with greater clarity and precision. Slowly, but surely, the tide began to turn. The identification of a protozoan parasite as the etiological agent of malaria, rather than a bacterium, presented a conceptual hurdle for many scientists. The scientific community was accustomed to identifying bacteria as the causative agents of infectious diseases, a consequence of the burgeoning field of bacteriology spearheaded by figures like Robert Koch. The concept of a protozoan parasite, a more complex and less understood type of microorganism, as the cause of malaria seemed implausible to many. Laveran's persistence, however, gradually began to erode the wall of skepticism. He continued to refine his methods, meticulously documenting his observations and presenting his findings with unwavering conviction. He understood that convincing the scientific community would require more than just his own testimony. It would require providing irrefutable evidence that others could independently verify. The increasing quality of available microscopes aided in this endeavor, allowing other researchers to examine blood samples with greater clarity and precision. Slowly, but surely, the tide began to turn. Initial resistance to Laveran's groundbreaking discovery stemmed from a confluence of factors, prominently including the limitations inherent in the microscopy technology of the late 19th century. While microscopes had advanced considerably, Achieving consistently clear and high-resolution images remained a challenge. Variations in lens quality, illumination techniques, and slide preparation methods could all introduce artifacts or obscure the delicate structures of the malarial parasite, making it difficult for other researchers to replicate Laveran's observations reliably. Furthermore, the ephemeral nature of the parasite's appearance in blood samples posed a significant obstacle. Laveran himself emphasized the importance of examining fresh blood immediately after its extraction from the patient. The parasite's morphology could change rapidly as the blood aged, leading to misidentification or rendering it entirely undetectable. This requirement for fresh samples created logistical difficulties, particularly for researchers working in locations distant from malarial regions. The challenges associated with transporting fresh blood samples, maintaining their integrity during transit, and ensuring their timely examination contributed to the inconsistent results reported by some of Laveran's contemporaries. The prevailing paradigm in infectious disease research also contributed to the initial skepticism surrounding Laveran's findings. During the late 9th century, bacteriology reigned supreme. Scientists had successfully identified bacteria as the causative agents of numerous diseases, including anthrax, tuberculosis, and cholera. This success led to a widespread belief that all infectious diseases were caused by bacteria. The notion that a protozoan parasite, a type of microorganism far less understood than bacteria, could be responsible for a disease as prevalent and devastating as malaria challenged this established paradigm. Prominent figures in the scientific community further complicated the acceptance of Laveran's work by proposing alternative explanations for the cause of malaria. Edwin Klebs, a renowned German pathologist, claimed to have discovered a bacillus, which he named Bacillus malariae as the true causative agent of the disease. Similarly, Corrado Tommaso Crudeli, an Italian physician, identified another bacillus, which he called Bacillus malariae as the culprit. These competing claims, supported by respected scientists, diverted attention from Laveran's observations and fueled the debate surrounding the etiology of malaria. Klebs's claim, presented with considerable authority, influenced many researchers to seek bacterial explanations for malaria. His stature in the scientific community lent credence to his findings, even though they ultimately proved to be erroneous. Tommaso Crudeli's work, similarly, garnered attention and contributed to the confusion surrounding the true cause of malaria. The existence of these alternative hypotheses, championed by established scientists, made it more difficult for Laveran to gain acceptance for his protozoan theory. Laveran directly addressed the problem of skepticism by meticulously documenting his observations and presenting his findings at scientific meetings. He understood that convincing the scientific community would require more than simple assertions. It would necessitate providing detailed descriptions and compelling evidence that others could independently verify. He meticulously recorded the morphology of the parasite at different stages of its development, noting its characteristic pigment granules and its movements within the red blood cells. He also carefully documented the clinical symptoms of malaria patients and correlated them with the presence of the parasite in their blood. At scientific meetings, Laveran presented his findings with clarity and conviction, using detailed illustrations and microscopic preparations to support his claims. He patiently answered questions from skeptical colleagues, addressing their concerns and providing additional evidence to bolster his arguments. He also published his observations in scientific journals, making them accessible to a wider audience and inviting scrutiny from other researchers. This commitment to transparency and rigorous documentation helped to gradually erode the initial skepticism surrounding his work. The increasing quality of available microscopes also played a crucial role in the eventual acceptance of Laveran's discovery. As microscope technology advanced, lenses became more powerful and image resolution improved. This allowed other researchers to examine blood samples with greater clarity and precision, making it easier to observe the malarial parasite and confirm Laveran's findings. The development of new staining techniques further enhanced the visibility of the parasite, making it even easier to identify in blood samples. Figures like Robert Koch, despite their initial focus on bacterial causes of disease, indirectly contributed to the eventual acceptance of Laveran's work by establishing rigorous standards for identifying and characterizing infectious agents. Koch's postulates, which outline the criteria for establishing a causal relationship between a microorganism and a disease, provided a framework for evaluating Laveran's claims. While Laveran's initial observations did not fully satisfy Koch's postulates, his meticulous documentation and the subsequent work of other researchers helped to gradually meet these criteria, strengthening the evidence for the protozoan etiology of malaria. The criticisms leveled against Laveran's initial findings directly influenced his subsequent research. The skepticism of his contemporaries forced him to refine his methods, improve his documentation, and seek additional evidence to support his claims. He recognized that simply observing the parasite was insufficient. He needed to demonstrate its causal relationship to malaria. He therefore focused on correlating the presence of the parasite with the clinical symptoms of the disease and on ruling out other potential causes. This rigorous approach, driven by the need to address the concerns of his critics, ultimately strengthened his case and contributed to the eventual acceptance of his discovery. The limitations of microscopy, the prevailing belief in bacterial causes, and competing claims from other researchers all contributed to the initial skepticism surrounding Laveran's discovery. His meticulous documentation, his unwavering conviction, and the increasing quality of available microscopes gradually convinced other scientists. The influence of figures like Koch, who established rigorous standards for identifying infectious agents, and directly supported Laveran's case. The criticisms from his contemporaries forced him to refine his methods and seek additional evidence, ultimately strengthening his arguments and paving the way for the eventual acceptance of his groundbreaking discovery. The initial resistance to Laveran's findings highlights the deeply entrenched scientific paradigms of the late 9th century. The prevailing belief in the bacterial origin of infectious diseases, championed by figures like Robert Koch, cast a long shadow. Laveran's observations, challenging this dominant view, demanded extraordinary evidence. Overcoming the skepticism required not only meticulous observation, but also independent verification and the development of techniques to make the parasite more visible. Camillo Golgi, a renowned Italian physician and histologist, played a pivotal role in validating Laveran's discovery. Golgi, celebrated for his innovative staining methods that revolutionized the study of the nervous system, turned his attention to the malarial parasite. Influenced by Laveran's initial reports, Golgi meticulously examined blood samples from patients suffering from malaria. His keen observations led him to describe the life cycle of the parasite within human red blood cells, a critical step in understanding the pathogenesis of the disease. Golgi identified distinct stages of parasitic development, correlating these stages with the characteristic fever cycles experienced by malaria patients. He observed, for instance, that the release of merozoites, a stage in the parasite's asexual reproduction, coincided with the onset of fever. This correlation provided a crucial link between the parasite's life cycle and the clinical manifestations of malaria. Golgi's detailed descriptions provided independent confirmation of Laveran's discovery. Golgi's work, in turn, influenced other researchers, such as Giovanni Battista Grassi, who further elaborated on the parasite's life cycle and its transmission. Golgi's contribution extends beyond mere confirmation. He identified two principal types of malaria, tertian and quartan, each associated with a specific species of Plasmodium. Tertian malaria, characterized by fever spikes every 48 hours, was linked to Plasmodium vivax. Quartan malaria, with fever spikes every 72 hours, was associated with Plasmodium malariae. This distinction, based on the parasite's morphology and developmental cycle, demonstrated that malaria was not a single disease entity, but rather a collection of related diseases caused by different, albeit related, parasites. This understanding had profound implications for diagnosis and treatment, paving the way for more targeted interventions. The visualization of the parasite also received a boost from the work of Richard Pfeiffer. Pfeiffer, a German physician and bacteriologist, developed improved staining techniques. These techniques enhanced the visibility of the malarial parasite under the microscope. His staining methods, employing aniline dyes, allowed researchers to more easily distinguish the parasite from the surrounding blood cells, thereby facilitating its identification and study. Pfeiffer's work directly aided researchers in replicating Laveran's observations. Scientists could now more readily visualize the parasite, bolstering the evidence supporting Laveran's claim. The discovery of different species of Plasmodium, coupled with improved staining techniques, gradually shifted scientific opinion. Initially met with skepticism, Laveran's discovery gained traction as more researchers were able to observe the parasite and confirm its association with malaria. The meticulous work of Golgi, the enhanced visualization provided by Pfeiffer, and the growing body of evidence linking the parasite to the disease gradually eroded the prevailing belief in bacterial causation. Laveran's initial discovery profoundly shaped Golgi's research trajectory. Prior to Laveran's work, Golgi's research focused primarily on histology and the nervous system. Laveran's identification of the malarial parasite as the causative agent of malaria provided Golgi with a specific target for investigation. This directed Golgi's attention to the study of blood samples from malaria patients. Laveran's influence is evident in Golgi's meticulous descriptions of the parasite's life cycle within red blood cells. Golgi focused on identifying different stages of development and correlating those stages with the fever cycles observed in patients. Without Laveran's groundbreaking discovery, Golgi's research would likely have taken a different path. Golgi's validation, in turn, exerted a substantial influence on the acceptance of Laveran's discovery. Golgi's reputation as a meticulous scientist and innovator lent considerable weight to his findings. His detailed descriptions of the parasite's life cycle and his identification of different species of Plasmodium provided compelling evidence supporting Laveran's claim. The scientific community, initially hesitant to embrace Laveran's discovery, gradually accepted it as valid, influenced by Golgi's endorsement. Scientists who had previously questioned Laveran's observations began to reevaluate their positions, swayed by the weight of Golgi's authority and the compelling nature of his evidence. Golgi's work served as a critical bridge, connecting Laveran's initial discovery to widespread scientific acceptance. The identification of distinct Plasmodium species and their association with different types of malaria provided a framework for understanding the diverse clinical presentations of the disease. This understanding had implications for diagnosis, treatment, and prevention. For instance, the knowledge that Plasmodium vivax could remain dormant in the liver, causing relapses, led to the development of drugs specifically targeting this dormant stage. Similarly, the recognition that different species exhibited varying degrees of drug resistance prompted the development of tailored treatment regimens. The identification of different species also facilitated the development of diagnostic tools, such as rapid diagnostic tests capable of distinguishing between different types of malaria. The shift in scientific opinion, driven by the accumulation of evidence and the influence of key figures like Golgi and Pfeiffer, marked a turning point in the understanding of malaria. The miasma theory, which had long dominated thinking about the cause of the disease, gradually gave way to the germ theory. The identification of the malarial parasite as a causative agent paved the way for a more rational approach to prevention and treatment. Scientists began to focus on understanding the parasite's biology, its life cycle, and its interactions with the human host. This focus ultimately led to the discovery of the mosquito vector and the development of effective control measures. Laveran's microscopic observations revealing the true cause of malaria initiated a paradigm shift in how the disease was understood and addressed. For centuries, the prevailing explanation for malaria's prevalence was a miasma theory, attributing illness to noxious vapors emanating from swamps and decaying organic matter. This theory, while attempting to explain the correlation between environmental conditions and disease incidence, offered little in the way of effective prevention or treatment strategies. Efforts focused on draining swamps and avoiding night air, measures that were often impractical and ultimately ineffective. The discovery of the malaria parasite by Laveran challenged this long-held belief. His meticulous examination of blood samples, revealing the presence of a microscopic organism, provided concrete evidence that malaria was caused by a living agent, a parasite invading the red blood cells. This discovery served as a cornerstone for the germ theory of disease, which posited that infectious diseases are caused by microorganisms. While the germ theory had been gaining traction, Laveran's work provided a compelling example of its applicability to a widespread and devastating illness. The implications of this paradigm shift were profound. The focus of research shifted from environmental factors to the parasite itself. Scientists began to investigate the parasite's life cycle, its mode of transmission, and its interactions with the human host. This new direction of inquiry ultimately led to groundbreaking discoveries that revolutionized the understanding and treatment of malaria. A direct consequence of Laveran's work was the impetus it provided for the eventual discovery of the mosquito vector by Ronald Ross. Ross, a British medical officer working in India, was directly influenced by Laveran's findings. Laveran's demonstration that malaria was caused by a parasite circulating in the blood inspired Ross to search for the parasite in mosquitoes, which were suspected of playing a role in the disease's transmission. Ross meticulously dissected mosquitoes that had fed on malaria patients, searching for evidence of the parasite's development within the insect. After painstaking investigation, Ross made the crucial observation that malaria parasites underwent a developmental stage within the Anopheles mosquito. This discovery, published in 1097, established the mosquito as the vector responsible for transmitting malaria to humans. Ross's work built directly upon Laveran's initial discovery, providing the missing link in understanding the complete life cycle of the malaria parasite. Ross himself acknowledged the profound influence of Laveran's work on his own research, recognizing that Laveran's identification of the parasite was the essential first step in unraveling the mystery of malaria transmission. Ross's discovery of the mosquito vector, directly informed by Laveran's identification of the parasite, opened new avenues for malaria prevention and control. Understanding that malaria was transmitted by mosquitoes allowed for the development of targeted interventions to interrupt the transmission cycle. Mosquito control measures, such as draining breeding sites, using insecticides, and implementing mosquito nets, became central to malaria prevention efforts. These strategies, based on the understanding of the parasite's life cycle in both humans and mosquitoes, proved to be far more effective than previous approaches based on the miasma theory. The identification of the mosquito vector also paved the way for a more complete understanding of the parasite's complex life cycle. Scientists discovered that the malaria parasite undergoes different stages of development in both the mosquito and the human host. In the mosquito, the parasite multiplies sexually, producing sporozoites that migrate to the salivary glands. When the mosquito bites a human, it injects these sporozoites into the bloodstream, initiating the infection. In the human host, the parasite undergoes asexual reproduction in the liver and red blood cells, causing the symptoms of malaria. This detailed understanding of the parasite's life cycle provided crucial insights for the development of new drugs and vaccines. Drugs could be targeted to specific stages of the parasite's development, disrupting its ability to multiply and cause disease. For example, drugs like quinine and chloroquine, which were initially derived from natural sources, were found to be effective in killing the parasite in the blood. The development of synthetic antimalarial drugs, such as artemisinin-based combination therapies, further enhanced the effectiveness of malaria treatment. Furthermore, the identification of specific proteins on the surface of the parasite offered potential targets for vaccine development. Vaccines could be designed to stimulate the immune system to recognize and attack the parasite, preventing infection or reducing the severity of the disease. While the development of an effective malaria vaccine has proven to be a challenging endeavor, significant progress has been made in recent years with the development of promising vaccine candidates that offer partial protection against malaria. This work is a direct continuation of the line of research that Laveran initiated. The problem Laveran solved was the identification of the true causative agent of malaria. This identification allowed for a shift from ineffective theories to targeted interventions. Prior to his discovery, efforts to combat malaria were largely based on speculation and conjecture. The identification of the parasite as the cause of malaria provided a rational basis for developing effective treatments and prevention strategies. The understanding that malaria was caused by a parasite circulating in the blood rather than by miasmas directly influenced future medical entomologists and public health officials. This understanding led to the development of targeted interventions to interrupt the transmission cycle, such as mosquito control measures and drug development. Medical entomologists focused on studying the behavior and ecology of mosquitoes to identify effective methods for controlling their populations. Public health officials implemented large-scale programs to distribute mosquito nets, spray insecticides, and provide access to antimalarial drugs. These efforts, guided by the understanding of the parasite's biology and mode of transmission, have significantly reduced the burden of malaria in many parts of the world. The legacy of Laveran's discovery extends far beyond the immediate impact on malaria control. His work played a pivotal role in establishing the germ theory of disease and revolutionizing the approach to understanding and combating infectious diseases. His meticulous observations and persistence in the face of skepticism serve as an inspiration to researchers today. The insights gained from his work continue to inform modern research on parasitic diseases and contribute to the ongoing efforts to improve global health. His work directly influenced future medical entomologists and public health officials, whose efforts, guided by the understanding of the parasite's biology and mode of transmission, have significantly reduced the burden of malaria in many parts of the world. Alphonse Laveran's enduring legacy resides in his profound impact on parasitology and the broader understanding of infectious diseases. His identification of Plasmodium as the causative agent of malaria marked a paradigm shift, moving away from prevailing miasmatic theories and solidifying the germ theory of disease. This discovery provided a concrete target for intervention, stimulating research into the parasite's life cycle, transmission mechanisms, and potential vulnerabilities. Laveran's contribution extended beyond identification. It shaped the very methodology of parasitological investigation. His meticulous microscopic observations, detailed descriptions of the parasite's morphology at various stages of its development, and its persistence in the face of initial skepticism established a rigorous standard for future researchers. He demonstrated the importance of direct observation and careful documentation in unraveling the complexities of parasitic infections. The Nobel Prize in Physiology, or medicine, awarded to Laveran in 1907 served as a powerful validation of his groundbreaking work. The Nobel Committee recognized the transformative nature of his discovery, acknowledging its profound implications for the understanding, treatment, and prevention of malaria. This prestigious award not only honored Laveran's individual achievement, but also elevated the field of parasitology, bringing it into the mainstream of biomedical research. His work provided a foundation for subsequent investigations into the life cycle of malaria parasites. Researchers like Ronald Ross, inspired by Laveran's initial discovery, painstakingly traced the development of Plasmodium within the Anopheles mosquito, definitively establishing the role of mosquitoes in malaria transmission. Ross's work, directly building upon Laveran's identification of the parasite, earned him the Nobel Prize in 1902 further solidifying the significance of Laveran's initial breakthrough. The impact of Laveran's work reverberates through modern malaria research. Contemporary efforts to develop new antimalarial drugs, vaccines, and diagnostic tools are all predicated on the fundamental understanding of the parasite that Laveran first provided. The ongoing quest to eradicate malaria relies on strategies that target specific stages of the parasite's life cycle, disrupt its transmission, and prevent its development within the human host, all informed by the knowledge that originated with Laveran's observations. Consider the development of artemisinin-based combination therapies, or ACT, the current mainstay of malaria treatment. These drugs, derived from the Artemisia annua plant, target specific biochemical pathways within the parasite, disrupting its growth and replication. The effectiveness of ACTs hinges on a precise understanding of the parasite's metabolism, knowledge that stems directly from the groundwork laid by Laveran and his successors. Similarly, vaccine development efforts focus on identifying parasite antigens that can elicit a protective immune response in humans. These antigens, molecules on the surface of the parasite, are carefully selected based on their ability to stimulate the production of antibodies that neutralize the parasite or prevent it from infecting liver or red blood cells. The identification and characterization of these antigens rely on sophisticated molecular techniques, but the underlying principle remains the same, targeting the parasite that Laveran first visualized under his microscope. Laveran's influence extends beyond malaria, shaping the broader field of parasitology. His discovery served as a template for investigating other parasitic diseases, inspiring researchers to search for the causative agents of illnesses such as trypanosome RCs, leishmania RCs, and schistosome RCs. The methods he pioneered, careful observation, microscopic examination, and experimental investigation, became standard practice in parasitological research. His work served as a catalyst for the development of new staining techniques and microscopic methods that allowed researchers to visualize parasites more clearly and to study their morphology in greater detail. Fumanoski stains, for example, commonly used in hematology and parasitology, enable the differentiation of various cellular components, facilitating the identification of parasites within blood samples. These techniques, developed in response to the need to visualize and study parasites, have become indispensable tools in diagnostic laboratories worldwide. Laveran's meticulous approach to scientific inquiry, his unwavering dedication to his research, and his courage to challenge prevailing theories serve as an enduring source of inspiration for scientists today. His story exemplifies the power of observation, the importance of persistence, and the transformative potential of scientific discovery. He demonstrated that even the most entrenched dogmas can be overturned by careful experimentation and a willingness to embrace new evidence. His legacy is further cemented by the inclusion of his work in modern parasitology curricula. Students around the world learn about Laveran's discovery as a foundational moment in the history of medicine, recognizing its significance in shaping our understanding of infectious diseases. Textbooks routinely feature images of Plasmodium parasites, often accompanied by historical accounts of Laveran's groundbreaking observations. His influence is also evident in the structure of modern parasitology research. Laboratories dedicated to the study of parasitic diseases routinely employ techniques and methodologies that trace their origins back to Laveran's work. Researchers continue to build upon his foundations, exploring the intricacies of parasite biology, developing new diagnostic tools, and searching for innovative ways to combat parasitic infections. Consider the application of genomics and protonics to the study of parasitic diseases. These cutting-edge technologies allow researchers to analyze the entire genetic makeup and protein complement of parasites, providing unprecedented insights into their biology and pathogenesis. While these techniques represent a significant advance over Laveran's microscopic observations, they are ultimately aimed at achieving the same goal, understanding the parasite in order to develop effective interventions. Laveran's work, therefore, represents then a historical milestone. It is a living legacy that continues to shape the field of parasitology and to inform the ongoing fight against infectious diseases. His discovery of the malaria parasite revolutionized medicine, providing a concrete target for intervention and paving the way for the development of effective treatments and prevention strategies. His meticulous observations and unwavering dedication serve as an inspiration to researchers today, reminding us of the power of scientific inquiry to transform our understanding of the world and to improve the health of humanity. His influence is woven into the fabric of modern parasitology, ensuring that his name will forever be associated with the quest to conquer parasitic diseases.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.